Hi, I'm Kyle Kim, RA here at Countable Labs, and I want to share with you today a really cool DNA linkage analysis assay for cell and gene therapy applications using Countable PCR. So the accurate characterization of complete versus partial transgene packaging in viral vectors is super critical for the efficacy and safety of gene therapy products, and it's often one of the biggest challenges in the cell and gene therapy space. Countable PCR makes DNA linkage assays more straightforward than in digital PCR due to two key features. The first being single molecule isolation in a 3D matrix, which enables you to get counts with unmatched precision. And then two, the use of our universal multiplexing probes, which allows you to design and optimize your assay in a matter of days rather than the weeks or months it may take with digital PCR and TaqMan probes. So let me show you an example in which I targeted three regions of a transgene. So here in this experiment, I targeted the CMV region, a GFP reporter gene, and a poly A tail of a vector plasmid. Universal multiplexing uses adapters on standard PCR primers to tag target amplicons with your probes, making it more affordable and faster to iterate than with TaqMan probes. Countable Labs single molecule isolation in a 3D matrix with over 30 million compartments also makes characterizing complete versus partial transgene packaging so accurate. And how does it do this? Well, um, this 3D matrix ensures that each molecule is being occupied in a single compartment, and amplification of that compartment gives you information on what target or targets may be present in that single molecule. If two or more targets are present in that molecule, they would be considered to be linked. So here in this scheme, I would expect channels 1, 2, and 3, or blue, green, and orange, to represent a complete transgene. In the instances where the transgene is not fully packaged or incomplete, I would expect a mixture of partial signals. And so here, this is just an output of our countable PCR system, in which the three different light sheets represent the three different targets of our assay. And so you can see here that the system automatically overlays the images, quantifies, and runs the DNA linkage analysis. And so here, when we would have only one or two targets present in the compartment, you would get this partial signal look. So when you have a compartment that is orange, we would only expect that molecule to contain a GFP and a poly A portion. And in a partition or compartment that is blue, we would expect that molecule to only have the CMV enhancer present. And in these compartments where you have a white light, that's where all three signals are overlaid together. And so that would represent you have a complete transgene with all three components. So to summarize, from generating the initial primer design to receiving the final data, this whole process took me only about 3 days to finish. And so our universal multiplexing kit makes it super easy to interrogate multiple targets, and only uses standard PCR primers to target your regions of interest. Additionally, because our system does the counting and linkage analysis automatically, there's no need for elaborate bioinformatics or manual thresholding to receive your counts for each channel. At the end of the run, you simply get an output for the counts for each channel, as well as information on what fraction they co-occupy. If you're interested in running DNA linkage analysis or easy multiplexing experiments, please feel free to reach out to one of our specialists at Countable Labs today.